Question and answer session for a short time after this next talk. But Neil Harrison from the University of Sussex is going to return us to a topic that we talked about a bit last year, but things have moved on a long way since then. And this is brain imaging in relation to chronic fatigue syndrome and ME, and particularly some of the nice work going on in functional brain imaging. So, Neil, thank you.
Thank you, Stephen. Let me just turn this on and make sure you can hear me. Is that okay? Yeah, thank you very much, Stephen, for the invitation to speak and to give people an update on where we're at with one of the MRC-sponsored studies. So what I'm going to talk about and what I'm going to present is the study itself. We have some preliminary data on this study at the moment. So we've recruited 14 people with ME/CFS, not enormous numbers, I appreciate, at the moment, and 10 controls, but recruitment is going well now. And as you'll see when I sort of go through the slides, you'll see it's a pretty complicated study. It's not a completely straightforward study. But what we were particularly interested in, so this is a study that's a collaboration between Sussex and St George's Medical School in London, is this question about post-exertional malaise in ME/CFS. Can we devise an experimental way to try and tap into this, to try and find out what on earth is going on? And I'm sure everyone in the room is well aware of post-exertional malaise, but I suppose the critical point here really is that we don't really understand why post-exertional malaise exists. What causes it? What generates it? There is, I mean, I cribbed some of this actually from the ME Association website, but essentially there's a growing sort of belief based on evidence that ME/CFS is an illness triggered by pro-inflammatory changes. So, for example, many cases or many individuals who have ME/CFS report a previous preceding infective episode, and we know that when you experience an infection or any other stressor, you get a release of pro-inflammatory cytokines. These are just proteins that the immune system uses to communicate with itself and amongst itself. And this triggers flu-like symptoms and malaise. Or even in healthy individuals, if you give them cytokines, you can induce a range of different symptoms. And it's not just infection that triggers this immune response or this inflammatory response. You can trigger it with all sorts of stressors. And I was just wondering in the previous talk with the cryotherapy whether cryotherapy itself is perhaps generating an inflammatory type response. You can certainly generate it with exercise, for example. So if if I were to go on a treadmill or on an exercise bike, a number of my cytokines would increase quite rapidly and quite acutely following that challenge. You can also do it with cognitive challenges. So we've done various studies over the years where we stress people in the lab, healthy individuals, and we can see even in the placebo condition when they're not given an immune challenge, we see an increase in these pro-inflammatory cytokines. They're not enormous, they're not massive increases, but they are robust changes. So what our primary hypothesis here is really this, is that we predict that people with ME/CFS are going to show a heightened and more persistent inflammatory response to a stressor, and the stressor that we're using is an exercise stressor. And we predict that this increased inflammatory response will be revealed, or we will see evidence of this response in the brain imaging data that we're collecting at the same time. So this is sort of a summary slide, if you like, of what I've already been talking about. I mean, on the left-hand side here, if anyone was here last year, they'll have seen this slide before, but essentially this is a cartoon on the left-hand side here of what happens when the immune system runs into an infection or a challenge that it recognizes. Pattern recognition receptors on innate immune cells recognize elements on the infecting agent, they become activated, release cytokines, and they coordinate the immune system response. However, these cytokines also act on the brain, and there's a picture here from Edvard Munch just sort of illustrating what we already know. Whenever we get an infection, our behavior changes. We all know this. Whenever we get the flu, our behavior changes. And how do we know that it's cytokines? Well, probably a couple of decades of work in rodents have shown that if you take a rodent and you give it a pro-inflammatory cytokine, so not an infection, just one of these cytokines produced by the body, you can induce a whole host of behavioural changes that as a group are known as sickness behaviours, and they include things like fatigue, changes in motivation, so the effort exerted to gain rewards, a whole host of other cognitive changes, cognitive slowing, psychomotor slowing, so our actions become slower, and other things like hypersensitivity to pain. We can also do these sort of studies in humans. We can't give them individual cytokines, or rarely give them individual cytokines because they're potentially extremely toxic. But the studies that we've done, we've used things like lipopolysaccharide, which is, if you like, the cell wall of a bacteria that's purified. It's not an infection, but you can give it to healthy volunteers to activate their immune system. And we're doing that locally, and other groups are doing that around the world. You can also give people things like a typhoid vaccination, which is a very mild challenge. But in this study, we're going to use exercise as our challenge. So this is going to be, if you like, the stressor or the pro-inflammatory challenge in this study. And what do we know when you activate the immune system? What goes on? How does the brain ever know that you've had an infective episode? Well, there are a number of different pathways that have been worked out in rodents on the left, and also some of our sort of very early work trying to map out these pathways in humans. So we know that there are, for example, nerves, visceral afferent nerves, so these travel with nerves like the vagus nerve and sympathetic nerves that are sensory. They respond to pro-inflammatory cytokines in your body, they communicate to the brain, and we know that because we see increase in activity in projection areas in rodents, and also when we do imaging in humans, we see with fMRI, for example, activation of what we call an interceptive network. So a network, a very specific network, that is believed to monitor the physiological state of our body. Also, and we've shown this, and other groups have shown this as well with neuroimaging, that you activate this interceptive pathway, and this projects ultimately to this little bit of the brain that I've ringed in circles here, which is called the insula. So when you activate this, the highest point that this information travels to is cortex, up in the insular cortex. And we've used a variety of different imaging techniques, fMRI, PET, fluorodeoxyglucose PET, so this just looks at metabolism, and a more modern technique called QMT, which I'm not really going to talk about. But it doesn't really matter what the technique is you use. You see activation of this. You can also see it with different challenges. So we use typhoid in all of these studies, but other groups have used LPS and shown sort of similar things. And interestingly, if you take a bunch of healthy individuals, say 20, 30 healthy individuals, you give them an inflammatory challenge, and then you wait 3 hours and put them in the scanner, and you also ask them how fatigued they are. Very simple, just a visual analog scale from 0 to 100, how fatigued are you? And you look at that change after inflammation. We find that activity within this interoceptive pathway correlates with how fatigued they feel following the inflammation. So the more you activate the system, the more fatigued a healthy individual feels. And just the final little sort of intro slide here, really, this bit of the brain, this insular cortex, is a large piece of brain. It doesn't just worry about inflammation, it worries about every physiological change within your body, so acid-base balance, heart rate variability, a whole host of other information that's coming from inside your body, not the outside world, but from within your body. And these are just a whole host of other studies looking at things like thermoregulation, response to temperature, graded itch, exercise, for example, all of which show activation within this insular cortex. So what are we doing in this study then? So this is a schematic, and I'm going to go through this schematic in a little bit more detail in a moment. But essentially the study looks like this. There are 3 parts to the study. So there's an initial, what we call a pre-visit, and I'll go through in a bit more detail what we're doing at the pre-visit, where we screen people with ME/CFS to ensure that they're safe to take part in the study, that they fulfill criteria, and then they have 2 additional visits. The first visit, we take some blood samples, give them some questionnaires, do an MRI task, and then put them on an exercise bike. And I'll show you the details of this in coming slides. We take another blood sample after that exercise, and then they go home. Then the next day they come back again, and we repeat all of the questionnaires, blood sampling, and the MRI, and then repeat an exercise task again. So they get 2 exercise tasks, 2 MRI scans in this study. It's a relatively small study. There are going to be a total of 40 people involved in the study, each of them being scanned on 2 separate occasions, so a lot of data, 80 scanning and testing sessions. And we're collecting lots of different data. So on the MRI, I'll show you some of the things we're collecting. We're collecting information about their performance during the exercise task, and we're collecting blood to look at a variety of different things, individual cytokines, and also ultimately to look at transcriptomics, so look at what is changing globally in transcriptome within the blood. So this is just a little bit more detail about each of those 3 individual sort of periods. So the pre-visit, all of the people are recruited from the Sussex-wide MECFS service, and then we reassess them and ensure that everybody who comes into the study fulfills both FACUDA and Canadian criteria for MECFS. And then we complete a number of different questionnaires, including the DePaul post-exertional malaise questionnaires. That's one of the sort of the critical features in this study. We also do ask them questionnaires about interoception and interoceptive awareness. And what I mean by that is how good you are at labeling your bodily responses. And the tasks that we use on this, I'm not going to give you much more details on this, but essentially we can play a set of tones and ask people if these tones are in time with their heartbeat, or ask them how many heartbeats they experience in particular parts of time. And this is a fairly well validated and commonly used way of trying to quantify how good we are at reading our own bodily signals. On day 1, I've already mentioned this already, we do bloods at the beginning of the day, questionnaires, we do this interoceptive task, and then we do scanning and the exercise task. And then day 2 is the day after the previous scanning session, and again we repeat many of these tests, the MRI and the exercise task. So what is this exercise protocol that people are doing then? What is it that they're involving, what they're doing? And it's quite a major exercise task. So people come in, it's a very standardized task, and the aim is to safely reach each individual's maximum exercise capacity. So this is a task that's used in numerous other labs around the world, but you can see immediately when you see this slide that we're recruiting people that have relatively mild to perhaps moderate MECFS symptoms. This is clearly not a task that would be that many people with more severe MECFS will be able to complete. So they cycle on a stationary bike for about 8 to 12 minutes. And then every minute, the resistance on the bike gradually increases. Throughout all of this, there's a wide array of monitoring going on. So recording inhaled and exhaled gases, recording heart rate, recording a range of physiological parameters. We also record things like subjective exertion, so how much do you think you're exerting yourself on this task subjectively, and also subjective fatigue. And there are a number of different stopping criteria on here. We can stop when the participant says, look, I've had enough, that's it, or we can stop when the experimenter decides that maximal exercise capacity has been reached. Okay, so there are 2 different sort of endpoints potentially for this. And I just want to show you a picture of what this looks like. And this is not one of our MECFS participants. I think you can probably quickly see that, but this is an athlete from the Brighton Sesame School of Exercise Science, and he's volunteered to put on the rig and demonstrate for us, because I think it's sometimes easier to see with a picture exactly what's involved. But essentially it's on a bike, and you can see all of the gases are recorded. We record a collection of different things. So for example, on the bottom here we record how well people are responding to this task. We can look at their heart rate and monitor how they proceed up this sort of step ladder in time. We also record other things like the oxygen consumption, the carbon dioxide that they're breathing out, and the 2 questionnaires there are just looking at perceived exertion and fatigue. With the MRI scanning, we're doing a lot of different MRI scanning, and I'm not going to go into vast details on this unless somebody is a real sort of particular fan of MRI, and I'm very happy to talk at length about what we're doing here. But we're doing a task, a Stroop task that we've used before and that we've shown is sensitive to inflammatory challenges. This is a stressor type task. It's quite hard, and I'll show it to you in the next slide. The next one is resting state. So this is people just lay in the scanner for 15 to 20 minutes, and we look at how different parts of the brain are connected. we do some microstructural imaging, and then we do what's called diffusion imaging to look at how well the brain is structurally connected. And this is an example of the Stroop task. So I don't know if it's getting a little bit late in the day, but this is the task here. So this is the control condition. So what you need to do is choose the option on the bottom here that corresponds to the colour of that word at the top. So just yell out for me what colour font that word is. Perfect. Okay, as fast as you can, what's the colour font of that one?
Red.
All right, blue. Okay, where the blue came from. But okay. I mean, you've already had a little bit of time to think about this. But under pressure, it becomes quite difficult because what you want to say is green, and you have to inhibit this response before you say red. So this is quite stressful. And the way this is done in the scanner, you're under a degree of time pressure. So if you're doing well at this task, we speed it up. And if you're doing badly, then we slow it down. So we try and titrate to performance on this task. But essentially, this is quite a stressful, I find this quite a stressful task to do myself, and that's the whole purpose of this, to do it. And what we've shown is that when you give healthy individuals an inflammatory challenge, for them to maintain their performance on here, they have to start recruiting additional bits of the brain. So they have to start recruiting more of these frontal regions in order to maintain performance. So obviously the prediction here is that we would anticipate we'd see something similar in the ME/CFS group on the day after that they've had the exercise challenge. So do they need to recruit additional bits of the brain in order to maintain their performance on this task? Now, I haven't got any data to show you, so I'm just going to show you a single bit of data that we've got so far. This is the resting state data, and what we are going to be doing is analysing the resting state data in a fairly sophisticated manner using what's called the Human Connectome Project pipelines. The beauty of doing this, number one, is it's sort of really state-of-the-art, but number two is that any data collected from this study can then be put into a bioresource, if you like, an imaging bioresource, similar to what we've heard about earlier with the cellular bioresources, so that other people can add in. And this is, if you like, a standard now for how you do resting state data analyses across the world. So these data can be added on by additional groups who want to look at this. But essentially, all I'm showing you here at the moment is what's called a myelin map. So what we can produce is pictures, very pretty pictures, of bits of your brain that have more white matter in them. Okay, so which bits of the cortex have more white matter? And all you can see here is that this bit here is the motor cortex. So the bit of our brain that governs motor performance has more myelin in it than other bits of the brain. Why on earth is this so important? Well, what we ultimately want to do is to be able to extract for each individual the 300 different bits of the brain, specialized bits of the brain, that we believe are critical to human cognition. And the only way we can do that is by mapping this on an individual basis. So for example, I don't know, your motor cortex might be 10% bigger than mine, or it might be in a slightly different position. And if we don't take account of that, then any of the further analyses become a little bit meaningless.
Which is the front bit?
Front here.
Disorientated.
Front here, temporal lobes here. It's what's called a sort of an expanded map, if you like. So it's been smoothed out. And another very pretty picture, because again, I haven't got any data to show you on them at the moment, but there's another technique called diffusion imaging, which looks at the travelling of water throughout the brain. So water moves better along fibres than it does across fibres. So if you've got a neuron like this, water will move more quickly in this direction than it will across the fibres. And what we can do with this type of data is produce these rather beautiful maps looking at the structural connectivity of the brain. So again, this data will be collected. Again, this is collected in a uniform manner. And then we will begin to look to see whether there are disconnection differences between people with ME and controls. All right, so let's give you a few results. And these are totally preliminary results at the moment. And I just want to sort of illustrate, we've got 4 posters here for this. And this data is from Amy's, they're posters 1 to 4 at the back there. But this is just looking at exercise parameters at baseline. So on that first time when people come in, how do the ME/CFS group compare to controls? And as we've already heard this morning, I mean, there's already clear differences in a number of different parameters here. So people with ME/CFS are generally less good or have a lower total oxygen or maximal oxygen consumption. So they, if you like, they cannot use as much oxygen as healthy individuals. But there are a number of other interesting findings here. So for example, this one here on total time, and I'll go into this in a little bit more detail. So people with ME/CFS don't last as long on this task as control individuals. So that poses the next question, why? Why is it? Is it just because of the fatigue, or is there something else going on? And this is some data from Rae, again, poster at the back there. First off, just showing what I've already showed you. So this is the patient group on day 1 and day 2. You can see their maximum oxygen consumption is fairly similar at day 1 and day 2. So there seems to be a fair reliability of these findings. This is the control group, and you can clearly see that the control group are better able to use oxygen and to exercise at a higher level than the ME/CFS group. But this data I think is quite interesting, and this is looking at how long individuals can remain on this exercise bike at day 1 and day 2. So these are your control participants over here. You can see they stay on the bike longer than the ME/CFS group, but when you get to day 2, there's a sort of a slight increase in how much time they can stay on this bike, okay? But if you look at the ME/CFS group, you can see quite the opposite is happening. So you're getting a reduction in how long people can sustain their effort on the bike. And why is that? Well, and just bearing in mind, this interaction, there's only limited data in here, so this is a non-significant interaction at the moment. It's a trend, basically. But the interesting thing, I think, is that the reason that people aren't able to stay on here isn't, and this is the bit that's dropped off the bottom, because of how much they perceive they're exerting themselves, but it's because we're telling them to stop. So they're already hitting our predetermined stopping criteria. So from maximum predicted heart rate or the RER. So they're getting to a stage where we're saying enough, that's as much as we want you to exercise. So this is actually quite, the changes here are being driven by how much they're exerting themselves rather than any subjective reasoning. And then looking here, so these are the ratings of fatigue. So looking at how people rate fatigue at peak exercise on day 1 and on day 2. And you can see in the control group, there's a slight reduction in this purple line here of how fatigued they feel at maximum effort or maximum exercise in the control group. So they're feeling slightly less fatigued on day 2 than day 1. But of course, in the ME/CFS group, we're seeing quite the reverse sort of pattern that's happening here. This is a significant interaction, but again, I would caution these are early days. We've got about half the data in here at the moment. But there does seem to be a clear pattern here on ratings of fatigue. And there's one other set of data that we're exploring at the moment, and this is very provisional data, but we're trying to explore and get around this question of, is there something about how good we are at reading our own internal physiological state that somehow relates to how well we can perform on an exercise bike? Very early provisional data at the moment suggests that those people that are better able to read their bodily state, and there's massive variety in this. I mean, I've done this, I'm not terribly good at this myself, but there's enormous variety or variability between people. But what we're finding here at the moment is that those people that are better able to read their internal physiological state are the ones that are able to remove carbon dioxide from their body better. So if you like, they seem to be breathing out more carbon dioxide. Go and have a chat with Nick at his poster if you've got ideas about why and what this means and why this is the case. But again, provisional data, but I think this is kind of quite an interesting observation. And then there's another observation here looking at total exercise time. And again, what we find is those people that have better interoceptive ability are better able to continue for longer on the second day of testing. Again, I don't really want to say any more about that at the moment, but it would be good to perhaps have a chat with Nick if you get a chance. Why on earth are we seeing these sort of patterns emerge? Right, so there's 2 final slides, and this is just what else can we do with the data that we've collected. So we will be collecting transcriptomic data at baseline and then the day after they've had their exercise challenge. And of course, what we would quite like to know is, can we identify a pattern within what's being translated within the blood that correlates with an increase in fatigue from day 1 to day 2, or any of the other different sort of questionnaire type measures. And all I'm showing you here is some data from patients with hepatitis C. So this is some older, currently unpublished data from people with hepatitis C who were given interferon alpha. So they didn't get an exercise challenge, they were given a cytokine challenge. And you see a massive upregulation in all sorts of different genes being expressed within the blood, exactly as you would expect. You give someone a cytokine, you get massive increase in 500-600 different genes. And this is highly significant. This is highly significant, and this enrichment is well above what you'd expect by chance. But this isn't the reason that we did this. What we were interested in knowing is, can we predict which of these individuals who show a response at 4.5 hours after their first interferon go on to develop really quite severe fatigue 4 weeks later? So we monitored these people who are receiving this treatment over 6 months to see, can we predict at the 4.5 hour window what's going on 4 weeks later. And again, this is just some provisional, currently unpublished data, but there does seem to be an interesting pattern emerge. So those people that show the greatest increase in this blob, if you like, on the graph, which is actually a ubiquitination, a proteasome cluster, seem to be the ones that then go on to develop the most severe fatigue. So if you like, this is involved in the degradation of proteins. So those people that start degrading proteins acutely within 4.5 hours of interferon administration appear to be the ones that then go on to experience the most fatigue 4 weeks later. So this sort of analysis is what we will be planning to do with some of the transcriptomic data that comes in. So I'm going to leave it there. Thank you very much for your attention. A large number of people are involved with this. So Mahinda is a neurologist from St George's who's here at the back of the room. Nick Smeaton, Amy Allington and Ray Hawkins, who are involved with the exercise tasks, are sitting at the table in the middle, and I'm sure would love to chat to you afterwards. So thanks for your attention.